Good evening, everyone. From today onwards, Sankalp Board and KCET classes for academic year 2025 and 26 will be conducted daily from 6 o'clock to 7 o'clock. It is sponsored by Rotary Bangalore Midtown and Rotarian Suresh Bhatia sir. These classes are free of cost for every students. So I request all the students to take the advantage of these classes to get the good marks in your PU exams. Okay. Yes, this is the brief introduction about the Sankalp ACE program. Once again, welcome to all students for this Sankalp learning classes. Yes. Total 13 chapters are there in biology. So from today onwards, I'm going to start with the first chapter, that is sexual reproduction in flowering plants. Yes, this is the first chapter. So we are going to complete the total 13 chapters in this program. Okay, both board and the KCET classes. So let us start with the first chapter. So sexual reproduction in the flowering plants. So first thing is, what is sexual reproduction? Sexual reproduction means where there is a fusion of the male and female gametes takes place, right? So allenagutte male matte female gametes production agutte, and there is a fusion of the male and female gametes, so that produces the zygote, which is not available in the asexual reproduction. You know what is asexual reproduction? There is no formation of the gametes takes place, right? So flowering plants. So what is the meaning of flowering plants? Flowering plants means angiosperms. Okay. So these angiosperms, they are nothing but the flowering plants. So in this chapter, we are going to study about the sexual reproduction in the flowering plants. In angiosperms, the flower is the reproductive part. Okay. Yes, let us see the structure of the flower. Yes. So if you see the flower, mainly four important whorls we can see in the flower. The first one is the outermost green color structure, that is the sepal, and group of sepals that is called as calyx, right? And the colorful part of the flower, that is the corolla. So this is petal. Yes, this is the colorful part of the flower, that is the petal, group of petals, that is called as corolla. Okay. Colorful part, that is the corolla, and the male reproductive part and the female reproductive part. So this is the male reproductive part. So what it is called? So this is the stamen, that is the male reproductive part, and group of stamens, that is called as androecium. Okay. So group of stamen, that is called as androecium. Then the female reproductive part, that is the pistil. Yes, this is the, so just I will round up this. Then the female reproductive part anta karitare. So that is the pistil. And this pistil contain the stigma, style and the ovary. Alva, idr bagge ನೀವು already ಕಲ್ತ್ಕೊಂಡಿದ್ದೀರಾ ಅಂತ ಅನ್ಕೊಳ್ತೇನೆ. Pistil, that is the female reproductive part. And so this part, it is called as stigma, and this elongated part, that is called as style, and the bulged part that is known as ovary. That is the female reproductive structure. And this pistil, group of pistil, that is called as gynoecium. Androecium and gynoecium. Here, these are the very important structures of the flower. That is calyx, corolla, androecium, gynoecium. Okay. Here, the calyx and corolla. So these whorls are called as this calyx and corolla, or you can call it as sepal and petals. They're called as accessory whorls. Accessory whorls. Accessory whorls. And this androecium and the gynoecium, male reproductive part and the female reproductive part. So that is called as the necessary whorls. Okay, so these are the necessary whorls. Please remember this, accessory whorls and necessary whorls in the flower. Okay, so this is the structure of the flower. So in this chapter, in detail, we are going to study about the male reproductive structure, how the male gametes are produced, and how female gametes are produced, how the fusion of male and female gamete takes place. So these all things we are going to study in this chapter. Yes, pre-fertilization and structure and the events. Here, what is the meaning of pre-fertilization events? Before the fertilization, what is the meaning of fertilization? Fusion of the male and female gametes, right? So the fusion of the male and female gametes, that is called as fertilization. Pre-fertilization events means before the fertilization, what are the events takes place that we are going to study. That is, several hormonal and structural changes are initiated which led to the differentiation for the development of the floral primordium in a plant. So there is a development of the floral primordium takes place, and inflorescence are formed which bear the floral buds and then the flowers. So we have studied about the inflorescence in the first year classes, right? So in the flower, the male and female reproductive structures are the androecium and the gynoecium differentiates and develop. So the differentiation of the androecium, that is the male reproductive part, and the gynoecium, that is the female reproductive part. So the differentiation of these parts takes place in the pre-fertilization events. Okay, so let us study about the male reproductive structure. So that is the stamen. So here, so if you see the flower, I hope.
In this video, we will learn the parts of a flower and their functions.
Once again, watch the parts of the flower. So this is the outermost wall.
This part is called episepals. Sepals.
Sepals. And watch the petals.
Petals. Stamen.
And the male reproductive part. Stamens. These are the stamens, male reproductive structure. And this is the female reproductive structure.
Pistil.
Pistil. ನೋಡಿ pistil ಯಾವ ತರ ಇದೆ ಅಂತ. style ಮತ್ತೆ ovary ನ ನೋಡಬಹುದು ಅಲ್ವಾ. This white color structure, that is ovary, and this stalk like structure, that is style, and this is the stigma. The female reproductive structure. So next we are going to study about the male reproductive structure, that is stamen, microsporangium and the pollen grain. Okay. So if you see the male reproductive structure, so you can observe the anther. So anther is like this. ಸೊ ನೀವು ಹೈಬಿಸ್ಕಸ್ ದಾಸವಾಳ ಹೂವನ್ನ ನೋಡಿರ್ತೀರಾ ಅಲ್ವಾ, ಅಲ್ಲಿ ಯೆಲ್ಲೋ ಕಲರ್ ಒಂದು ಸ್ಟ್ರಕ್ಚರ್ ಅನ್ನ ನೀವು ನೋಡಿರ್ತೀರಾ ಅಲ್ಲಿ. ಸೊ ದಿಸ್ ಸ್ಟ್ರಕ್ಚರ್ ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಆಂಥರ್. ಸೊ ಆಂಥರ್ ನೋಡಿ, ಸೊ ವಿಚ್ ಹ್ಯಾವ್ ದ ಫಿಲಮೆಂಟ್. ಸೊ ದಿಸ್ ಸ್ಟಾಕ್ ಲೈಕ್ ಸ್ಟ್ರಕ್ಚರ್ ಅನ್ನ ಏನಂತ ಕರೀತಾರೆ. ಸೊ ದಿಸ್ ಇಸ್ ದ filament and this is the anther. So this is the main parts of the stamen. Together it is called as a stamen. Anther and the filament together it is called as a stamen. Okay, male reproductive structure. Anther ಮತ್ತೆ ಫಿಲಮೆಂಟ್ ಅನ್ನ ಜೊತೆಗೆ ಸೇರಿಸಿ ಸ್ಟೇಮನ್ ಅಂತ ಕರೀತಾರೆ ಅಲ್ವಾ. A typical stamen, a long slender stalk called, that is the filament, and terminal generally bilobed structure called as the anther. The terminal part which is having the bilobed structure, that is called as the anther. And the proximal end of the filament is attached to the thalamus or the petal of the flower. So this is the proximal end, this is the distal end alva. So the proximal, very important for your exam. Which part of the filament is attached to the thalamus? The proximal end. So this is the proximal end, this is the distal end. Proximal end is attached to the thalamus or the petal of the flower. So here a typical angiosperm anther is bilobed, which is having the two lobes. ಎರಡು lobes ಇದೆ. ನೀವು ಯಾವುದೇ anthers ಅನ್ನ ತೆಗೆದುಕೊಂಡ್ರೆ, so which is having the two lobes. Okay, so that is the bilobed structure. So you here you can observe the bilobed. ಇಲ್ಲಿ ಕಾಣಿಸ್ತಾ ಇದೆ ಅಲ್ವಾ, ಎರಡು lobes ಇದೆ. So it is called as the bilobed structure. Each lobe with having the two theca. If you take the section of this transverse section of this anther, so you can observe the structure like this. ನೋಡಿ ಈ ತರ ಸ್ಟ್ರಕ್ಚರ್ಸ್ ನಿಮ್ಗೆ ಕಾಣಿಸುತ್ತೆ. ಯಾವ ತರ ಇದೆ ನೋಡಿ, ಈ ರೀತಿಯಾಗಿದೆ ಅಲ್ವಾ. Yes, if you take the transverse section of this anther, you can observe the structure like this, and each lobes. So this is one first lobe, this is the second. So two lobes are there, right? So each lobe that is having the two theca. So this is the first and the second one, two theca. Hence it is called as the dithecous anther. So each lobe that is having the two theca. So this is the first lobe and this is the second lobe. Each lobe having the sorry, this is the first lobe, second lobe. Each lobe is having the two theca, hence it is called as the dithecous anther. So ನಿಮಗೆ ಎಕ್ಸಾಮ್ ಅಲ್ಲಿ ಕೇಳ್ತಾರೆ, a typical angiosperm anther is dash and dash. So that is the bilobed. So ಎರಡು lobes ಇರುತ್ತೆ, and if you take the section of this anther, you can observe the two theca in each lobe. Hence it is called as the dithecous. ಇಲ್ಲಿ ನೋಡಿ ಈ ತರ ಸ್ಟ್ರಕ್ಚರ್ ಇದೆ ಅಲ್ವಾ. So this is the first and this is the second. Two theca. You can observe the two theca, hence it is called as the dithecous anther. The anther is a four-sided, that is the tetragonal. If you see the transverse section of the anther, you can see the four-sided structure, that is called, what it is called, tetragonal, right? So which is having the tetragonal, four-sided alva, ಇಲ್ಲಿ ಕಾಣಿಸ್ತಾ ಇದೆ ಅಲ್ವಾ ನಿಮಗೆ, four-sided structure. So that is the tetragonal, and each consisting of microsporangia. So you can observe the round structure here. So that is nothing but the microsporangia. So ಈ microsporangia ಅಂದ್ರೆ ಏನು ಅಲ್ವಾ? So inside the microsporangia, microspores production ಆಗತ್ತೆ. What is this microspores? Microspores, they further develop into a pollen grains, male gametophyte ಅಲ್ವಾ? ಮೇಲ್ ಗ್ಯಾಮೇಟ್ ಅನ್ನ ರಿಲೀಸ್ ಮಾಡುವಂತ ಸ್ಟ್ರಕ್ಚರ್ಸ್ ಆಗಿ ಡೆವಲಪ್ ಆಗತ್ತೆ ನೋಡಿ. Anther, if you take the section of the anther, you can observe the microsporangia here. So these are the microsporangia, and this microsporangia, so they produces the microspores and further they develop into a pollen sacs. In microsporangia, they develop further become a pollen sacs. Idanna pollen sacs antanu kuda, so it will develop into a pollen sacs because at the maturation stage, so yavaga idu mature agatte alva anther, aga adu pollen grains which is filled with the pollen grains, hence it is called as a pollen sacs. Okay, so that is about the anther. So let us see the structure of microsporangium. Okay, so let us see the structure of the microsporangium. This diagram is very important for your board exam. Okay, so 5-mark question. Sorry. So when you take the section of the anther, you can see the tetragonal four-sided structure like this. Yes, so this is the structure. So what you observe. So here, this is the microsporangia. Let us see the structure of the microsporangium. So this microsporangium generally surrounded by 4 wall layers. So this microsporangium, that is surrounded by 4 wall layers. Here you can see the 4 wall layers. This is the outermost layer which is called as epidermis. Microsporangium ಅಲ್ಲಿ outermost layer ಅನ್ನ ಏನಂತ ಕರೀತಾರೆ? That is the epidermis. And the second layer, that is the endothecium. Layer. And the third layer, that is the middle layer. And this is the tapetum. Okay, so very important, which are the 4 wall layers of the microsporangium. The outermost layer is the epidermis, and next layer endothecium. Sorry, this is the epidermis, and second layer, that is the endothecium. Then this layer is the middle layer, then this is the tapetum. Okay, so these are the 4 wall layers of the microsporangium, and this is the microsporangia, and this one is the microsporangia, innermost microsporangia. This microsporangia, which is surrounded by the 4 wall layers. I hope you got this point. ಅರ್ಥ ಆಗ್ತಾ ಇದೆ ಅಂತ ಅನ್ಕೊಳ್ತೇನೆ. If you have any doubts, you can write down in the chat box. Okay. The outer 3 wall layer perform the function of the protection and help in the dehiscence of anther to release the pollen. So ಇಲ್ಲಿ 3 wall layers ಇದೆ ಅಲ್ವಾ? ಎಪಿಡರ್ಮಿಸ್ ಇದೆ, ಎಂಡೋಥೀಶಿಯಂ, ಮಿಡಲ್ ಲೇಯರ್. ಸೊ ದಿಸ್ ಆರ್ ದ ಪ್ರೊಟೆಕ್ಟಿವ್ ಲೇಯರ್. ಸೊ ಇದನ್ನ ಏನ್ ಮಾಡುತ್ತೆ? ಮೈಕ್ರೋಸ್ಪೊರಾಂಜಿಯಂಗೆ ಪ್ರೊಟೆಕ್ಷನ್ ಕೊಡುತ್ತೆ ಅಂಡ್ ಆಲ್ಸೋ ಹೆಲ್ಪ್ಫುಲ್ ಫಾರ್ ದ ಡಿಹೈಸೆನ್ಸ್ ಆಫ್ ದಿ ಆಂಥರ್. ಸೊ ಇಲ್ಲಿ ಈ ಒಂದು ಆಂಥರ್ ಡಿಹೈಸೆನ್ಸ್ ಆಫ್ ದಿ ಆಂಥರ್, ಇಲ್ಲಿ ಆಂಥರ್ ಬ್ರೇಕ್ ಡೌನ್ ಆಗಿ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ರಿಲೀಸ್ ಆಗಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ. And the innermost wall layer, which is the innermost wall layer, that is the tapetum, right? So this tapetum, very important, it nourishes the developing pollen grains. So pollen grains ಎಲ್ಲಿ ಪ್ರೊಡಕ್ಷನ್ ಆಗ್ತಾ ಇದೆ? ಮೈಕ್ರೋಸ್ಪೊರಾಂಜಿಯಾದಲ್ಲಿ ಪ್ರೊಡಕ್ಷನ್ ಆಗ್ತಾ ಇದೆ ಅಲ್ವಾ? The pollen grains, they are producing inside the microsporangia. And this tapetum that provides the nutrition for this developing pollen grains. ಇಲ್ಲಿ ಕೇಳ್ತಾರೆ, so the function of the tapetum that provides the which nourishes the developing pollen grains, and when the anther is young, the group of compactly arranged homogeneous cell layer called the sporogenous tissue occupies in the center of the microsporangium. ಸೊ ಇಲ್ಲಿ ಏನಿರುತ್ತೆ ಅಂದ್ರೆ ಈ ಮೈಕ್ರೋಸ್ಪೊರಾಂಜಿಯಾ ಒಳಗಡೆ ಸ್ಪೋರೋಜಿನಸ್ ಟಿಶ್ಯೂ ಇರುತ್ತೆ. So inside the microsporangia, there is a sporogenous tissue is present. Okay. Here, the tapetum, that is the multinucleated, so which is having the cytoplasm with the multinucleated. Okay, so ಇಲ್ಲಿ ಕೇಳ್ತಾರೆ tapetum which provides the nourishment for the developing pollen grains as well as which is the multinucleated structure. So multinucleate means which is having the more than one nucleus in the single cell. Okay, so that is the multinucleate. So tapetum, very important for your examination, and remaining three layers which are protective in function and help for the dehiscence of anther, and tapetum that nourishes the developing pollen grains, and that is multinucleate in structure. Okay, so inside the microsporangium, when the anther is young, the microsporangium that contains the sporogenous tissue. So these all words are new for you people. Sporogenous tissue is present. Next, so let us see the how the microspores are produced inside the microsporangium. So the process microsporogenesis. What is microsporogenesis? The process of formation of microspores from the pollen mother cell through the meiosis is called as a microsporogenesis. So how this process takes place? So we are going to study about this process in detail. Microsporogenesis andre ಏನಾಗತ್ತೆ ಅಲ್ವಾ? Microsporogenesis means microspores. So these all things refers to the male reproductive structure. Okay, micro ಯಾವಾಗಲೂ ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ, micro ಅಂದ್ರೆ male reproductive structure and the mega means female reproductive structures. Okay, so as the anther develops, the cells of the sporogenous tissue undergo meiotic divisions to form microspore tetrads. ಇಲ್ಲಿ ನೋಡಿ ಏನಾಗುತ್ತೆ, inside the microsporangium, so microsporangium ಅಲ್ಲಿ sporogenous tissue ಇರುತ್ತೆ ಅಲ್ವಾ? Sporogenous tissue, so that is diploid in structure. So diploid means that is 2n ಅಲ್ವಾ? So here the cells of the sporogenous tissue that will develop into a pollen mother cell. Pollen, PMC, pollen mother cell, that is diploid in structure. Okay, so that is diploid in nature. Sporogenous tissue that contains the pollen mother cell, that is mother cell. Then pitkoli mother cell means that is diploid. And this pollen mother cell that will undergo the meiosis. ಮಿಯೋಸಿಸ್ ಆದಾಗ ಏನಾಗುತ್ತೆ, ಪೋಲನ್ ಮದರ್ ಸೆಲ್ ಏನಾಗ್ತಾ ಇದೆ, ಮಿಯೋಸಿಸ್. ಸೊ ಯಾವಾಗ್ಲೂ ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಸೆಲ್ಸ್ ಅಲ್ಲಿ ಮಿಯೋಸಿಸ್ ಆಗುತ್ತೆ ಅಲ್ವಾ? ಸೊ ಡಿಪ್ಲಾಯ್ಡ್ ಇರೋದೇನಾಗತ್ತೆ, ಸೊ ವೆನ್ ಇಟ್ ಅಂಡರ್ಗೋ ದ ಮಿಯೋಸಿಸ್ 1 ಅಂಡ್ ಮಿಯೋಸಿಸ್ 2 ಅಲ್ವಾ? ಮಿಯೋಸಿಸ್ ಅಲ್ಲಿ 2 ಸ್ಟೆಪ್ ಇದೆ, ಮಿಯೋಸಿಸ್ 1 ಅಂಡ್ ಮಿಯೋಸಿಸ್ 2. ಸೊ ದಟ್ ಈಸ್ ಟೋಟಲಿ ಕಾಲ್ಡ್ ಮಿಯೋಸಿಸ್. ಸೊ ಇಟ್ ಪ್ರೊಡ್ಯೂಸಸ್ ದ 4 microspores. Okay, so pollen mother cell, or you can call it as microspore mother cell. Mother cell ಎಷ್ಟಿರುತ್ತೆ, diploid in condition ಇರುತ್ತೆ. So where it is present, inside the microsporangia ಅಲ್ವಾ? ಈ microsporangia ಒಳಗಡೆ ಎಲ್ಲ microsporogenesis ನಡೀತಾ ಇದೆ. ಅರ್ಥ ಆಗ್ತಿದ್ಯಾ? And which produces the microspores, 4 microspores. So they are haploid ಅಲ್ವಾ? ಯಾವಾಗ ಮಿಯೋಸಿಸ್ ಆಗಿ ನ್ಯೂ ಸೆಲ್ಸ್ ಫಾರ್ಮೇಶನ್ ಆಗುತ್ತೋ ಆಗ ಏನಾಗಿರುತ್ತೆ, ದ ನ್ಯೂ ಸೆಲ್ಸ್ ದೇ ಆರ್ ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಇನ್ ನೇಚರ್, ದಟ್ ಈಸ್ n in number. So here these haploid cells, so these 4 microspores, they are together. They are held together in a cluster of 4 cells. So these 4 cells are, these 4 microspores are, they are together in a cluster that is called as microspore tetrad. ಅರ್ಥ ಆಗ್ತಿದ್ಯಾ? Here, these microspores, as they formed, are arranged in a cluster of 4 cells. So that is called as microspore tetrad. Okay. So how the microspores are produced? That you know from the pollen mother cell. Right. As the anther mature and dehydrate, the microspores dissociate from each other and develop into a pollen grains. So when the anthers become mature, so in the young anther these all things are produced, and so when the anther get mature, so the and dehydrate, the microspores, they dissociate each other. So microspore ಏನಾಗಿತ್ತು, 4 microspores, they are tetrad in nature ಅಲ್ವಾ, 4 microspores together arrange ಆಗಿತ್ತು tetrad ತರ ಅಲ್ವಾ. So they dissociate each other. So these all microspores, they release from each other and they develop into a pollen grains, and the microspores, they develop into a pollen grains. Pollen grains ಅಂದ್ರೆ ನೀವು ನೋಡಿರ್ತೀರಾ, ಯೆಲ್ಲೋ ಕಲರ್ ಸ್ಟ್ರಕ್ಚರ್ ನಿಮ್ಮ ಕೈಗೆ ತಾಗತ್ತಲ್ಲ, ನೀವು ಯಾವುದೋ ಒಂದು ಫ್ಲವರ್ ಮುಟ್ಟಿದಿರಾ ಅಂತ ಅನ್ಕೊಳ್ಳಿ, ಆ ಫ್ಲವರ್ ಅಲ್ಲಿ ಯೆಲ್ಲೋ ಕಲರ್ ಪೌಡರ್ ಲೈಕ್ ಸ್ಟ್ರಕ್ಚರ್ ಇರುತ್ತೆ ಅಲ್ವಾ, ದಟ್ ಈಸ್ ನಥಿಂಗ್ ಬಟ್ ದ pollen grain. Okay. And these pollen grains, the microspores, they develop into a pollen grain. So this process, that is known as a microsporogenesis. So I hope you are getting. Then yes, so this is the mature anther. Nodi illi young anther irbekadre enagittu illi, so inside the microsporangia, idu microsporangia anta ankoli. So these all are microsporangia. Okay. So this microsporangia, so there is a microsporogenesis takes place. So where the microspores are produced inside the microsporangia alva. Microsporangia ಒಳಗಡೆ ಏನಾಗಿತ್ತು, microspores production ಆಗಿತ್ತು, but they are held together in a cluster of 4 microspores, that is called as a microspore tetrad. So ಈಗ ನೋಡಿ anther mature ಆಗ್ತಾ ಇದೆ, ಇಲ್ಲಿ ನೋಡಿ dehiscence anthers walls break down agi dehiscence, this is called dehiscence of anther. ಇಲ್ಲಿ ನೋಡಿ 4 wall layers ಇದೆ ಅಲ್ವಾ, ಎಪಿಡರ್ಮಿಸ್, ಎಂಡೋಥೀಶಿಯಂ, ಮಿಡಲ್ ಲೇಯರ್, ಟೆಪೆಟಮ್ ಎಲ್ಲ ಏನಾಗಿದೆ, ಬ್ರೇಕ್ ಆಗಿದೆ, ಇಲ್ಲಿ ಕಾಣಿಸ್ತಾ ಇದೆ ಅಲ್ವಾ. ಸೊ ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ dehiscence of anther. So then this microspores, they dissociate. 4 microspores ಇತ್ತು ಅಲ್ವಾ, so they dissociate each other and they are releasing from the, they develop into a pollen grains and they are releasing from the microsporangia. So the pollen grains, they are releasing from the microsporangia after the dehiscence of anther. Dehiscence of anther ಅಂದ್ರೆ ಇಲ್ಲಿ ನೋಡಿ, ಇಲ್ಲಿ ವಾಲ್ ಲೇಯರ್ಸ್ ಬ್ರೇಕ್ ಆಗಿ ಮೈಕ್ರೋಸ್ಪೊರಾಂಜಿಯಾದಿಂದ ಏನಾಗ್ತಾ ಇದೆ, ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ರಿಲೀಸ್ ಆಗ್ತಾ ಇದೆ ಅಲ್ವಾ. ಈ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ರಿಲೀಸ್ ಆಗ್ತಾ ಇದೆ. ಸೊ ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಅ dehiscence of anther. So in the mature anther it will takes place. And this microsporangium ಅನ್ನ ಏನಂತ ಕರೆದಿದ್ರು, pollen sac ಅಂತ ಕರೆದಿದ್ರು ಅಲ್ವಾ. Why it is called as a pollen sac? Because this microsporangium is filled with the this microsporangium is filled with the pollen grains ಅಲ್ವಾ. ಮೈಕ್ರೋಸ್ಪೋರ್ಸ್ ಏನಾಗಿತ್ತು, pollen grains ಆಗಿ ಡೆವಲಪ್ ಆಗಿ ಮೈಕ್ರೋಸ್ಪೊರಾಂಜಿಯಾದಲ್ಲಿ pollen grains ಇತ್ತು. ಸೊ ಲೇಟರ್ ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಅ pollen sac. Okay, so this is how the pollen grains are released. Okay, so pollen grains release ಆಗಿದೆ. ಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಸ್ಟ್ರಕ್ಚರ್ ಇಂದ ಏನಾಗಿದೆ, pollen grains. pollen grains ಅಂದ್ರೆ ನೀವು ನೋಡಿರ್ತೀರಾ, ನಾನು ಆಲ್ರೆಡಿ ಹೇಳಿರೋ ಹಾಗೆ ಯೆಲ್ಲೋ ಕಲರ್ ಸ್ಟ್ರಕ್ಚರ್, ನಿಮ್ ಒಂದು ಫ್ಲವರ್ ಅನ್ನ ಮುಟ್ಟಿದ್ರೆ ಅಲ್ಲಿ ಯೆಲ್ಲೋ ಕಲರ್ ಪೌಡರ್ ಲೈಕ್ ಸ್ಟ್ರಕ್ಚರ್ ನಿಮ್ಮ ಕೈಗೆ ಅಂಟ್ಕೊಳ್ಳುತ್ತೆ ಅಲ್ವಾ. ಇಫ್ ಯು ಟೇಕ್ ಹೈಬಿಸ್ಕಸ್ ಆರ್ ಎನಿ ಫ್ಲವರ್, ಸೊ ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಅ pollen grain. Then, so the pollen grains represents the male gametophytes, not male gametes. So they are male gametophytes. Okay, so they release the, they produces the male gametes. The pollen grains, they produces the male gametes, hence they are called male gametophytes. Okay, ಸೊ ನೋಡಿ ಪೋಲನ್ ಗ್ರೇನ್ ಯಾವ ತರ ಇರುತ್ತೆ. ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಆರ್ ಜನರಲಿ ಸ್ಪೆರಿಕಲ್, ಮೆಜರಿಂಗ್ ಅಬೌಟ್ 25 to 50 micrometer in diameter. Very important. 25 to 50 micrometer in diameter. And this pollen grain, so if you observe the pollen grain under the microscope ಅಲ್ಲಿ observe ಮಾಡಿದ್ರೆ you can observe the two wall layers in the pollen grain. Pollen grain ಅಲ್ಲಿ ನೀವು ಎರಡು wall layers ಅನ್ನ observe ಮಾಡಬಹುದು. The hard outer layer called exine is made up of sporopollenin, which is one of the most resistant organic material known. ನೋಡಿ ಇದೊಂದು pollen grain ಅಂತ ಅನ್ಕೊಂಡ್ರೆ the outermost layer. So just imagine this is the outermost layer. This outermost layer that is made up of the this is the pollen grain. So two layers ಇದೆ ಅಲ್ವಾ pollen grains ಅಲ್ಲಿ two wall layers ಇದೆ. And outermost layer of the pollen grain that is made up of the sporopollenin, which is the hardest or the most resistant organic material known. And inner layer is made up of outer layer is called as exine, made up of sporopollenin, and inner layer, so this is the exine, outer layer. Okay, this is the inner layer that is called as intine. Exine and the intine. And this intine that is made up of the cellulose and the pectin. Okay, here, so the sporopollenin withstand high temperature and strong acids and alkali. No enzyme that degrade the sporopollenin is so far known. Pollen grain exine has the prominent aperture called germ pores where the sporopollenin is absent. So nodi, so in the pollen grains you can observe the outermost layer exine, which is made up of the chemical sporopollenin alva, and innermost layer that is called as the intine. So this exine, so the sporopollenin that is very hard substance, very resistant substance, which is not degraded by the enzyme, acids, alkali. So ಯಾವುದು ಇದನ್ನ degrade ಮಾಡೋಕೆ ಆಗೋದಿಲ್ಲ. So that must resistant organic material it is. Okay, so but in the pollen grains, in some regions, the sporopollenin is absent. Sporopollenin absent irutte. So that is called as germ pore. Okay, so why it is necessary we will study later. So nodi idondu pollen grain andre, so ee ರೀತಿಯಾಗಿ pores irutte. So where the sporopollenin is absent. Okay, ನಾನು just ನಿಮ್ಗೆ ಅರ್ಥ ಆಗ್ಲಿ ಅಂತ ಬಿಡಿಸ್ತಾ ಇರೋದು. ನೋಡಿ ಇಲ್ಲಿ sporopollenin ಏನಿದೆ absent ಇದೆ ಅಲ್ವಾ. So that is called as a germ pore. ಕೇಳ್ತಾರೆ CET ಅಲ್ಲಿ ಕೇಳಿದ್ದಾರೆ. Where the exine where the sporopollenin is absent. So what it is called? That is called as a germ pores where the sporopollenin is absent. Okay. Yes, this is the pollen grain. This is the development of the pollen grain, how the maturation of the pollen grains takes place. Okay, the inner wall pollen grain is called intine, and which is made up of the cellulose and the pectin, which is the continuous layer. Okay, the inner layer is the continuous layer, but outer layer exine that is not continuous because germ pores are present alva. ನಾನು ಹೇಳಿದೆ germ pores irutte, outer layer ಅಲ್ಲಿ ಏನಿರುತ್ತೆ germ pores irutte. So where the sporopollenin is absent. The cytoplasm of the pollen grain is surrounded by the plasma membrane. When the pollen grain is mature, it contains two cells, the vegetative cell and the generative cell. When the pollen grain is mature, so this is not a mature pollen grain, this is the young pollen grain. Okay, so how the young pollen grain that will become mature? So a step ಅನ್ನಲ್ಲಿ ಕೊಟ್ಟಿದ್ದಾರೆ ನೋಡಿ. The nucleus that divides, that will form the two cells, that is vegetative cell and the generative cell. So vegetative cell and the generative cells are present in the mature pollen grain. So this is the ಮೆಚೂರ್ ಪೋಲನ್ ಗ್ರೇನ್. ಮೆಚೂರ್ ಪೋಲನ್ ಗ್ರೇನ್ ಯಾವ ರೀತಿಯಾಗಿ ಡೆವಲಪ್ ಆಗುತ್ತೆ, ಇಲ್ಲಿ 2 ಸೆಲ್ಸ್ ಆಗುತ್ತೆ ಅಲ್ವಾ, ನ್ಯೂಕ್ಲಿಯಸ್ ಡಿವೈಡ್ ಆಗಿ 2 ಸೆಲ್ಸ್ ಫಾರ್ಮೇಶನ್ ಆಗುತ್ತೆ. ಒಂದು ಪೋಲನ್ ಗ್ರೇನ್ ಅಲ್ಲಿ ಎಷ್ಟು ಸೆಲ್ಸ್ ಇರುತ್ತೆ? ಸೊ ವೆನ್ ಇಟ್ ಈಸ್ ಇನ್ ಎ ಮೆಚೂರ್ ಸ್ಟೇಜ್, ದಟ್ ಈಸ್ ದ vegetative cell and the generative cells are present, and the generative cell is small and floats in the cytoplasm of the vegetative cell. Nodi illi generative cell ide alva, so that is very small and which is compared to the vegetative cell. It is spindle shaped with dense cytoplasm and the nucleus. In over the 60% of angiosperms, pollen grain are shed at the 2-celled stage. Pollen grains 2-celled stage alli shed agutte. So and the generative cell in the remaining 40% species. So what happens? The generative cell divides mitotically to give rise to the 2 male gametes before the pollen grains are shed. So that is 3-celled stage. ಈ generative cells function ಏನು? Generative cell, so that produces the male gamete. Okay, generative cell that produces the male gametes and vegetative cell that provides the nutrition. Okay, so that is the function of the vegetative cell and the ಜನರೇಟಿವ್ ಸೆಲ್. ತುಂಬಾ ರಿಪೀಟೆಡ್ ಆಗಿ ಇದ್ರ ಮೇಲೆ ಕ್ವಶನ್ ಕೇಳಿದ್ದಾರೆ. ವೆಜಿಟೇಟಿವ್ ಸೆಲ್, ಜನರೇಟಿವ್ ಸೆಲ್, ಎಕ್ಸೈನ್, ಇಂಟೈನ್ ಅಂಡ್ ದೀಸ್ ಆಲ್ ಥಿಂಗ್ಸ್, ಜಂಪ್, ಇದ್ರ ಮೇಲೆ ಎಲ್ಲ ನಿಮ್ಗೆ ನಿಮ್ಮ ಬೋರ್ಡ್ ಎಕ್ಸಾಮ್ ಅಲ್ಲಿ ಇರ್ಬಹುದು, CET ಅಲ್ಲಿ ಇರ್ಬಹುದು, ತುಂಬಾ ಕ್ವಶನ್ಸ್ ಅನ್ನ ಕೇಳಿದ್ದಾರೆ. Next, what are the economic importance of the pollen grain? Okay, so pollen grains, you know that they cause the severe allergies, bronchial affections, some applications, some people often leading to the chronic respiratory disorders, asthma, bronchitis. So ಬೇರೆ ಬೇರೆ diseases ಅನ್ನ cause ಮಾಡುತ್ತೆ or allergies ಅನ್ನ cause ಮಾಡುತ್ತೆ. And pollen grains are rich in nutrients. It has become fashion in recent years to use the pollen tablets as a supplement. Okay, so pollen tablets ಅನ್ನ food supplement ಆಗಿ ಯೂಸ್ ಮಾಡ್ತಾ ಇದ್ದಾರೆ. In Western countries, the large number of pollen products in the form of tablets and syrups are available in markets. ನೋಡಿ ಇಲ್ಲಿ ಚಿತ್ರದಲ್ಲಿ ನೀವು ನೋಡಬಹುದು, ಟ್ಯಾಬ್ಲೆಟ್ಸ್ ಇರಬಹುದು, ಸಿರಪ್ಸ್ ಇರಬಹುದು. ಸೊ ಅದನ್ನೆಲ್ಲ ವೆಸ್ಟರ್ನ್ ಕಂಟ್ರೀಸ್ ಅಲ್ಲಿ ಯೂಸ್ ಮಾಡ್ತಾರೆ. ಅಂಡ್ pollen consumption has been claimed to increase the performance of athletes and race horses. So when the pollen grains shed, once they are shed, pollen grains have to land on the stigma. ಯಾವಾಗ pollen grains shed ಆಗಿದೆ, so that should land on the stigma ಅಲ್ವಾ? So that is the male gametophyte. ಅದೇನಾಗಬೇಕು, pollen grains ಏನಾಗಬೇಕು, that should transfer to the stigma. ಸ್ಟಿಗ್ಮಾಕ್ಕೆ ಏನಾಗಬೇಕು, just imagine this is the stigma. So that should land on the stigma. So before they lose the viability, so they have to transfer to the stigma. So stigma ge transfer ಆದ್ಮೇಲೆ the period for which the pollen grains remain viable is highly variable and to some extent depends on the prevailing temperature and the humidity. So ಈಗ stigma ಕ್ಕೆ transfer ಆಗಿದೆ. What is the viability of this pollen grains? So ಎಷ್ಟೊತ್ತು ಹಾಗೆ ಇರುತ್ತೆ, ವಯಬಿಲಿಟಿ ಎಷ್ಟು ಅದ್ರದ್ದು ಅಲ್ವಾ? ಸೊ ದಟ್ ಈಸ್ ಡಿಪೆಂಡಿಂಗ್ ಆನ್ ದಿ ಟೆಂಪರೇಚರ್ ಅಂಡ್ ದ ಹ್ಯೂಮಿಡಿಟಿ. ಸೊ ಇಟ್ ಈಸ್ ಡಿಫ್ರೆಂಟ್ ಇನ್ ಡಿಫ್ರೆಂಟ್ ಸ್ಪೀಶೀಸ್. ಬೇರೆ ಬೇರೆ ಸ್ಪೀಶೀಸ್ ಅಲ್ಲಿ ಬೇರೆ ಬೇರೆ ಟೈಮಿಂಗ್ಸ್ ಇದೆ. ಇನ್ ಸಮ್ ಸೀರಿಯಲ್ಸ್ ಸಚ್ ಆಸ್ ರೈಸ್, wheat, pollen grains lose viability within the 30 minutes of their release, and some members of Rosaceae, Leguminosae and Solanaceae, they maintain the viability for months. ನೋಡಿ ಈಗ ಸ್ಟಿಗ್ಮಾಕ್ಕೆ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗಿದೆ, 30 ಮಿನಿಟ್ಸ್ ಅಲ್ಲಿ ಇದು ಪೋಲನ್ ಟ್ಯೂಬ್ ಅನ್ನ ಜನರೇಟ್ ಮಾಡಿ, ಸೊ ದಟ್ ವಿಲ್ ರಿಲೀಸ್ ದ ಗ್ಯಾಮೇಟ್ಸ್ ಅಂಡ್ ಫ್ಯೂಸಸ್ ವಿತ್ ದ ಫಿಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಫಿಮೇಲ್ ಗ್ಯಾಮೇಟ್. ಸೊ ಓನ್ಲಿ 30 ಮಿನಿಟ್ ವಯಬಿಲಿಟಿ ಇದೆ. ಸೊ some members of the Rosaceae, Leguminosae and Solanaceae, they maintain the viability for months. So ತಿಂಗಳಾನುಗಟ್ಲೆ ಏನಾಗಿರುತ್ತೆ, so they have the viability. It is possible to store the pollen grains of large number of species for years in the liquid nitrogen minus 196 degrees Celsius. Okay, so that is possible to store the pollen grains in the liquid nitrogen. So the temperature is minus 196 degrees Celsius. Okay, so please remember this number, minus 196 degrees Celsius. And that pollen grains that is used for the breeding programs. Here, this is also very important. The members of Rosaceae, Leguminosae and Solanaceae, they have the viability for months. Okay, they will ask. So one question is asked in exam. So that is based on this. Please note down this. Then, so this is how the male gamete is produced. Pollen grains ಅಲ್ಲಿ generatives ಅಲ್ಲಿ ಏನ್ ಮಾಡುತ್ತೆ, ಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸ್ ಅನ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುತ್ತೆ ಅಲ್ವಾ. ಸೊ ವಿ ಹ್ಯಾವ್ ಕಂಪ್ಲೀಟೆಡ್ ವಿತ್ ದ ಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಸ್ಟ್ರಕ್ಚರ್. ಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಸ್ಟ್ರಕ್ಚರ್ ಯಾವ ರೀತಿ ಇರುತ್ತೆ, ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಹೇಗೆ ರಿಲೀಸ್ ಆಗುತ್ತೆ ಅಂಡ್ ಹೌ ದ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ರಿಲೀಸ್ ದ ಗ್ಯಾಮೆಟ್ಸ್. ಸೊ ದಟ್ ವಿ ಹ್ಯಾವ್ ಸ್ಟಡಿಡ್ ಇನ್ ದಿಸ್. ಸೊ ನೆಕ್ಸ್ಟ್, the pistil, megasporangium and the embryo sac. Okay, so the pistil means, so that contains the stigma, style and the ovary alva. Sorry, stigma, this is the stigma, and style and the ovary. Together it is called as a pistil, right? Together it is called as a pistil. The stigma serves as a landing platform for pollen grains. So pollen grains release ಆಗಿರೋದು ಎಲ್ಲಿ land ಆಗುತ್ತೆ, pollen grains they land on the stigma. So that is the landing platform where the pollen grains are received. Okay, the gynoecium represents the female reproductive part of the flower. The gynoecium may consist of single pistil. So nodi, gynoecium, the female reproductive structure, that may contain the single pistil irbodu alva, onde pistil irbodu. So just imagine this is the single pistil. Okay, so this is the stigma. Single pistil that is called as a monocarpellary. What is monocarpellary? That is that contain the single pistil and or may more than one pistil. So ಈಗ ಒಂದು ಫ್ಲವರ್ ಅಲ್ಲಿ ಒಂದೇ ಪಿಸ್ಟಿಲ್ ಇರಬಹುದು or more than one pistil ಇರಬಹುದು, 5, 10, 15 ಎಷ್ಟು ಇರಬಹುದು ಅಲ್ವಾ. If more than one pistils are present, that is multicarpellary. Just imagine ಒಂದು ಫ್ಲವರ್ ಅಲ್ಲಿ ಒಂದೇ ಪಿಸ್ಟಿಲ್ ಇರೋದಿಲ್ಲ, ಒಂದು ಪಿಸ್ಟಿಲ್ ಇರಬಹುದು ಅಥವಾ ಜಾಸ್ತಿನೂ ಇರಬಹುದು. Okay, that is called as multicarpellary. Very important. Monocarpellary and multicarpellary. When there are more than one pistils may be fused together, that is called as syncarpus. this is the syncarpus condition. ಇಲ್ಲಿ ಕಾಣಿಸ್ತಾ ಇದೆ ಅಲ್ವಾ. So here stigma, style and ovary. So ಇಲ್ಲೇನಾಗಿದೆ, ಮೋರ್ ದನ್ ಒನ್ ಪಿಸ್ಟಿಲ್ಸ್ ಏನಾಗಿದೆ, ಫ್ಯೂಸ್ ಆಗ್ಬಿಟ್ಟಿದೆ ಈ ರೀತಿಯಾಗಿ. ಪಿಸ್ಟಿಲ್ಸ್ ಸೆಪರೇಟ್ ಪಿಸ್ಟಿಲ್ಸ್ ಈ ರೀತಿ ಇದೆ ಅನ್ಕೊಡಿ, ಇದೆಲ್ಲ ಈ ತರ ಫ್ಯೂಸ್ ಆಗಿದೆ. ಸೊ ದಟ್ ಫ್ಯೂಸ್ಡ್ ಪಿಸ್ಟಿಲ್ಸ್ ಆರ್ ಕಾಲ್ಡ್ ಸಿಂಕಾರ್ಪಸ್. ಓಕೆ, ಸೊ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್, ಸಿಂಕಾರ್ಪಸ್ ಆರ್ ಮೇ ಬಿ ಫ್ರೀ, ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಅಪೋಕಾರ್ಪಸ್. ಯು ಕ್ಯಾನ್ ಸಿ ದಿಸ್ ಅಪೋಕಾರ್ಪಸ್, where the stigma, style and ovary pistils they are separate. More than one ide, onde illa, more than one ide, but they are not fused. That is called as a apocarpus condition. Inside the ovary, there is a ovarian cavity, locule and the placenta which is located inside the ovarian cavity. Okay, so ovule ಒಳಗಡೆ ಏನಿರುತ್ತೆ, ovarian cavity ಇರುತ್ತೆ where the ovules are located. Okay, so that is the that is called as pistil. Then the number of ovules in the ovary may be 1, so that is in wheat, paddy and mango, to many. So that is in papaya, watermelon and the orchids. So ovules number may, so that is differ in the different species. Let us see the structure of the megasporangium, that is the ovule. So ovule that is present inside the ovary alva. Ovary ಒಳಗಡೆ ಏನಿರುತ್ತೆ ovule ಇರುತ್ತೆ. So that you know. So that is called as ovule, that is also called as megasporangium. Draw a neat label diagram of the megasporangium or draw a neat label diagram of the ovule. So ಈ ರೀತಿಯಾಗಿ ಕೇಳ್ತಾರೆ ovule or megasporangium which is present inside the ovary alva. The ovule is the small structure attached to the placenta by means of stalk that is called as a funicle. So, ಇಲ್ಲಿ funicle, so where the stalk-like structure, which is the stalk-like structure, which is attached to the placenta. So, that is called as a funicle. Very important. ಫಿನಿಕಲ್. ಸೊ ಇಲ್ಲಿ ಡಯಾಗ್ರಾಮ್ ಅನ್ನ ನಿಮ್ಮ ಬೋರ್ಡ್ ಎಕ್ಸಾಮಿಗೆ ಹೇಳೋದಾದ್ರೆ ಡಯಾಗ್ರಾಮ್ ಅನ್ನ ಕೇಳ್ತಾರೆ ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್ ಆಗಿ. The body of the ovule fuses with the funicle in the region called hilum. So this is the body of the ovule. So that here is the funicle. That funicle that fuses with the body of the ovule, that region is called as the hilum. Okay. So that region is called as the hilum. Hilum represents the junction between the ovule and the funicle. So hilum is the junction between the ovule and the funicle. Funicle is the stalk-like structure. Okay. Each ovule that is covered by the protective structure that is called as the integuments. So ovule that is covered by the integuments, the protective layers that is called as the integuments, and there are two integuments, that is outer integuments and the inner integuments. Okay. Integuments encircle the nucellus. So this is the nucellus. So this is the nucellus part, and this nucellus part that is covered by the integuments. Nucellus except the tip of where a small opening is called micropyle is organized. Where the integuments are absent, you can see the structure here. Illi nodi, illi integuments absent ide. In the ovule where the integuments are absent, that is called as micropyle. Where the small region where the integuments are absent, that is the micropyle region. Okay. So illi integuments absent irutte. Then the opposite side of the micropyle, that is called as the chalazal region. Opposite of micropylar end, that is called as chalaza. So this is the chalazal end. Enclosed within the integuments in a mass of cells called nucellus. So integuments ಒಳಗಡೆ ಏನಿರುತ್ತೆ, there are nucellus is present which contains the mass of cells. Okay. ಇಲ್ಲಿ nucellus ಇರುತ್ತೆ. ಈ integuments ಒಳಗಡೆ ಏನಿರುತ್ತೆ, nucellus ಇರುತ್ತೆ. Cells of the nucellus have abundant reserve food materials. Located in the nucellus is the embryo sac or female gametophyte, and ovule generally has a single embryo sac formed from the megaspore. Okay. So ಇಲ್ಲಿ nucellus ಒಳಗಡೆ ಎಂಬ್ರಿಯೋ ಆರ್ ಫಿಮೇಲ್ ಗೆಮೆಟೋಫೈಟ್ ಯಾವ ರೀತಿಯಾಗಿ ಡೆವಲಪ್ ಆಗುತ್ತೆ ಅನ್ನೋದನ್ನ ನಾವು ನೋಡೋಣ. ಸೊ ಹಿಯರ್ ದಿಸ್ ಇಸ್ ದ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ದಿ ಮೆಗಾಸ್ಪೋರಾಂಜಿಯಂ. ಇದನ್ನ ನಿಮ್ಗೆ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ಲಿಕ್ಕೆ ಕೇಳ್ತಾರೆ. ಎಕ್ಸ್ಪ್ಲೇನ್ ದ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ದಿ ಮೆಗಾಸ್ಪೋರಾಂಜಿಯಂ ಆರ್ ಡ್ರಾ ದ ನೀಟ್ ಲೇಬಲ್ ಡಯಾಗ್ರಾಮ್ ಆಫ್ ದಿ ಓವ್ಯೂಲ್ ಆರ್ ಮೆಗಾಸ್ಪೋರಾಂಜಿಯಂ. ಈ ರೀತಿಯಾಗೂ ಕೂಡ ನಿಮ್ಗೆ ಕ್ವಶನ್ಸ್ ಅನ್ನ ಕೇಳ್ತಾರೆ. ನೆಕ್ಸ್ಟ್ ಮೆಗಾಸ್ಪೋರೋಜೆನೆಸಿಸ್. ವಾಟ್ ಈಸ್ ಮೈಕ್ರೋಸ್ಪೋರೋಜೆನೆಸಿಸ್ ಸ್ಟಡಿ ಮಾಡಿದ್ವಿ ಅಲ್ವಾ. What is megasporogenesis? The process of formation of megaspores from the megaspore mother cell is called megasporogenesis. So ಇಲ್ಲಿ megaspore mother cell ಎಲ್ಲಿರುತ್ತೆ ಅಲ್ವಾ. ಸೊ ಯಾವ ರೀತಿಯಾಗಿ megaspores production ಆಗತ್ತೆ. So these all things we have to study now. Okay. Ovules generally differentiate a single megaspore mother cell in the micropylar region of the nucellus. It is large cell containing the dense cytoplasm and the prominent nucleus. The megaspore mother cell undergoes the meiotic division. Nodi, the megaspore mother cell that is present inside the nucellus, that is the mass of cells alva. So nucellus ಅಲ್ಲಿ ಏನಿರುತ್ತೆ, megaspore mother cell ಇರುತ್ತೆ. Just imagine. So megaspore mother cell that is present inside the nucellus. Mother cell ಅಂದ್ರೆ ನಾನು ಹೇಳಿದ್ದೆ megaspore mother cell, microspore mother cell. So ಈ nucellus ಅಲ್ಲಿ ಇರುವಂತಹ megaspore mother cell that is diploid in condition ಅಲ್ವಾ. So megaspore mother cell that is diploid in condition and which undergo the meiosis. ಇಲ್ಲಿ ನೋಡಿ. So this is the megaspore mother cell. ಎಲ್ಲಿದೆ? So that is present inside the nucellus, and this will undergo the meiotic division and form the megaspores. Meiotic division, diploid ಇರುವಂತ ಸೆಲ್ meiotic division ಆಯ್ತು ಅಂದ್ರೆ ಎಷ್ಟು cells formation ಆಗುತ್ತೆ? 4 cells. So these are called as megaspores. Okay, so these cells are called as megaspores. See here, ಇಲ್ಲಿ micropylar end ಅಲ್ಲಿ ಅಂತ ಇದೆ ಅಲ್ವಾ. So these are the nucellus. So this is the megaspore mother cell. ನೋಡಿ, megaspore mother cell develop ಆಗಿದೆ, and this megaspore mother cell that will undergo the meiosis. So this is the megaspore mother cell that will undergo the meiosis 1. megaspore dyad ಅಂದ್ರೆ 2. dyad means 2 cells formation ಆಗಿದೆ. Then when it completes the meiosis, it forms the 4 megaspores ಅಲ್ವಾ. 4 megaspores ಅನ್ನ formation ಮಾಡಿದೆ ಅಲ್ವಾ. So from the megaspore mother cell. So that is called as megasporogenesis. megasporogenesis ಹೇಗಾಗಿದೆ? From the megaspore mother cell. megaspore mother cell, so that is present inside the nucellus, and that will undergo the meiosis and form the megaspores. 4 meiosis ಅಂದ್ರೆ ನಿಮಗೆ ಗೊತ್ತಿದೆ. meiosis ಅಂದ್ರೆ ಏನಾಗುತ್ತೆ? So one cell that is going to produce the total 4 cells ಅಲ್ವಾ. 4 cells production ಆಗುತ್ತೆ at the end of the meiosis. So here 4 megaspores are produced. Yes, you can see how the so this is the microsporogenesis, sorry. So let us see how the female gametophyte, that is embryo sac development takes place. Female gametophyte, embryo sac development. Okay. In majority of flowering plants, one of the megaspores is functional while the others are degenerate. ನೋಡಿ, microsporogenesis ಅಲ್ಲಿ ಏನಾಗಿತ್ತು? 4 microspores are present and they will develop into pollen grains, right? So ಒಂದು microspore mother cell ಇಂದ ಏನಾಗಿತ್ತು? 4 microspores production ಆಗಿತ್ತು ಅಲ್ವಾ. But here also from the megaspore mother cell that produces the 4 megaspores, but 3 of them will degenerate and only 1 will become functional. Very important. So during the formation of megaspores, 4 megaspores are produced, but only 1 will become functional. So only the functional megaspore that develops into female gametophyte. Male gametophyte, that is the ಯಾವುದು? pollen grain ಅಲ್ವಾ. pollen grain ಹೇಗೆ ಡೆವಲಪ್ ಆಗಿತ್ತು? microspores ಇಂದ pollen grain develop ಆಗಿತ್ತು ಅಲ್ವಾ. Here the megaspore that develop into a female gametophyte. What is female gametophyte? That is nothing but the embryo sac. Female gametophyte, that is nothing but the embryo sac. This method of embryo sac formation from the single megaspore is termed as monosporic development of the embryo sac. Very important. So only 1 megaspore develop into a female gametophyte, hence it is called as the monosporic development of the embryo sac ಅಂತ ಕರೀತಾರೆ. Here, so just I will explain through the diagram. So this is the megaspore. Okay, just imagine this is the megaspore, which is the functional megaspore. Okay, so this functional megaspore that will undergo the nuclear division ಆಗುತ್ತೆ, and that will become 2 nucleate stage. The megaspore which is formed by the ಮೆಗಾಸ್ಪೋರೋಜೆನೆಸಿಸ್ ಅಲ್ವಾ, ಒಂದೇ ಒಂದು ಮೆಗಾಸ್ಪೋರ್ ಉಳ್ಕೊಂಡಿದೆ ಅಲ್ವಾ, ಆ ಮೆಗಾಸ್ಪೋರ್ ಏನಾಗತ್ತೆ, ಸೊ ದಟ್ ವಿಲ್ ಅಂಡರ್ಗೋ ದ ಮೈಟೋಟಿಕ್ ಸೆಲ್ ಡಿವಿಷನ್. ಮೈಟೋಟಿಕ್ ಡಿವಿಷನ್ ಆಗತ್ತೆ, ಮೈಟೋಟಿಕ್ ನ್ಯೂಕ್ಲಿಯರ್ ಡಿವಿಷನ್ ಆಗತ್ತೆ. ಸೊ ಏನಾಗತ್ತೆ, 2 ನ್ಯೂಕ್ಲಿಯಸ್ ವಿಲ್ ಬಿ ಫಾರ್ಮ್ which will undergo the 2 nucleate stage. ನೋಡಿ, the nucleus of the functional megaspore divides mitotically to form the 2 nuclei, so that move to the opposite pole and forming the 2 nucleate embryo sac. ನೋಡಿ ಇಲ್ಲೇನಾಗತ್ತೆ, the nucleus of the, this is the nucleus of the functional megaspore and which will undergo the mitotic division and forms the 2 nucleus. ಎರಡು ನ್ಯೂಕ್ಲಿಯಸ್ ನೋಡಿ ಅಪೋಸಿಟ್ ಪೋಲ್ಸ್ ಹೋಗಿದೆ. ಸೊ ದಿಸ್ is called as a 2 nucleate embryo sac. And further again it will undergo the mitotic division and forms the 4 nucleate embryo sac, and further it will develop into a 8 nucleate. Only nuclear division takes place ಅಲ್ವಾ, ನ್ಯೂಕ್ಲಿಯಸ್ ಡಿವೈಡ್ ಆಗ್ತಾ ಇದೆ, ಈಚ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಡಿವೈಡ್ ಆಗಿ ಏನಾಗ್ತಾ ಇದೆ, 8 ನ್ಯೂಕ್ಲಿಯೇಟ್ ಕಾಣಿಸ್ತಾ ಇದೆ ಅಲ್ವಾ. ಇದು ಮತ್ತೆ ಡಿವೈಡ್ ಆಗಿದೆ, 8 ಆಗಿದೆ ಮೈಟೋಟಿಕ್ ಡಿವಿಷನ್ ಅಲ್ಲಿ. ಮೈಟೋಟಿಕ್ ಡಿವಿಷನ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, 1 ಇರೋ ಸಲ ಏನಾಗುತ್ತೆ, 2 ಆಗಿ ಡಿವೈಡ್ ಆಗುತ್ತೆ ಅಲ್ವಾ. ಸೊ ಈ ರೀತಿಯಾಗಿ ಏನಾಗಿದೆ, 8 ನ್ಯೂಕ್ಲಿಯೇಟ್ ಸ್ಟೇಜ್ ಆಗಿದೆ. ಆಫ್ಟರ್ ದ 8 ನ್ಯೂಕ್ಲಿಯೇಟ್ ಸ್ಟೇಜ್ there is a arrangement of this nucleus and cells takes place. Okay, arrangement of this inside the cell takes place. ಇಲ್ಲಿ ನೋಡಿ, ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ, so after the 8 nucleate stage, so that will become, so there is arrangement of the characteristic distribution of the cells takes place. So 3 cells are grouped together at the micropolar end and constitute the egg apparatus. ನೋಡಿ ಇಲ್ಲಿ, so this is the so here 3 cells. So 3 cells ಏನಾಗ್ತಾ ಇದೆ micropolar end ಅಲ್ಲಿ. So this is the micropolar end. So 3 cells, 3 nucleus are held together. ಸೊ ಇಲ್ಲಿ 3 ಏನಾಗಿದೆ, 3 ಸೆಲ್ಸ್ ಏನಾಗಿದೆ, ಸೊ ಜೊತೆಗೆ ಸೇರ್ಕೊಂಡಿದೆ ಅಲ್ವಾ. ಕ್ಯಾರೆಕ್ಟರಿಸ್ಟಿಕ್ ಡಿಸ್ಟ್ರಿಬ್ಯೂಷನ್ ಆಫ್ ಸೆಲ್ಸ್ ಟೇಕ್ಸ್ ಪ್ಲೇಸ್. 3 cells are grouped together at the micropolar end that constitute the egg apparatus. In the egg apparatus in turn consists of 2 synergids and 1 egg cell. So egg apparatus ಅಂದ್ರೆ ಏನಿದೆ, so 2 synergids are there. So ಇಲ್ಲಿ ನೋಡಿ, 2 synergids are there and the 1 egg cell ಇದೆ. So that is the egg cell, that is the female gamete alva. Egg apparatus that consists of 2 synergids and 1 egg cell is present. And again at a chalazal end ಅಲ್ಲಿ ಏನಾಗಿದೆ ನೋಡಿ, the synergids have the special cellular thickenings at the micropolar tip called piliform apparatus, which play the important role in guiding the pollen tubes into the synergid. So synergid, so they contain the cellular thickenings that is called as piliform apparatus. What is the function of the piliform apparatus? So that guides the pollen grains, okay, sorry, male gametes towards the egg cell. And 3 cells are at the chalazal end and they are called as antipodal cells. Okay, so here ಮತ್ತೆ ಉಳಿದಿರೋ 3 cells ಏನಾಗ್ತಾ ಇದೆ ನೋಡಿ. So these 3 cells, they are at the chalazal end. So they are grouped together and they are called as antipodals. ನೋಡಿ, antipodals ಅಂತ ಕರೀತಾರೆ. 3 cells micropolar end ಗಿದೆ, ಅದನ್ನ synergids and the egg apparatus, totally egg apparatus ಅಂತ ಕರೆದ್ರು. And chalazal end ಇರೋದನ್ನ ಏನಂತ ಕರೆದ್ರು, that is the antipodal cells ಅಂತ ಕರೆದ್ರು ಅಲ್ವಾ. So total 6 cells ಆಯ್ತು, total 8 nucleate stage ಆಗಿದೆ ಅಲ್ವಾ. So then the large central cell, so centrally ಏನಾಗತ್ತೆ ಅಂದ್ರೆ, so there is 2 nucleus, so that is present in the central ಅಲ್ಲಿ ಎರಡು ಸೆಲ್ ಇರುತ್ತೆ ಅದನ್ನ ಸೆಂಟ್ರಲ್ ಸೆಲ್ ಅಂತ ಸಾರಿ ಟೂ ನ್ಯೂಕ್ಲಿಯೇಟ್ ಸೆಲ್ ಇಸ್ ಪ್ರೆಸೆಂಟ್ ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಅ central cell. Okay, so here this is the structure of the matured embryo sac. So that contains the antipodals, synergids and the central cell. So totally this embryo sac is called as 8 nucleate 7 celled stage ಅಂತ ಕರೀತಾರೆ ಇದನ್ನ 8 nucleate 7 celled stage ಅಂತ ಯಾಕೆ ಕರೀತಾರೆ total 8 nucleus is present but 7 cells because central cell ಅಲ್ಲಿ so totally this is having the 2 nucleus in a single cell ಇಲ್ಲಿ 3 cells ಇದೆ ಇಲ್ಲೂ ಕೂಡ 3 cells ಇದೆ 6 cells ಆಯ್ತು ಅಲ್ವಾ so ಇಲ್ಲಿ ಒಂದು cell ಇದೆ but it contained the 2 nucleus. Okay, so total nucleus 8 but how many cells? 7 cells. So matured embryo sac that is called as 8 nucleate 7 celled stage. Okay, so at the maturity the embryo that is having the embryo sac that is having the 8 nucleate and the 7 celled stage. So this is how the female gametophyte is formed. Okay, so in the next class, so in today's class we have studied about the male reproductive structure and the microsporogenesis alva microsporogenesis ಯಾವ ರೀತಿಯಾಗಿ ಆಗುತ್ತೆ microspores ಯಾವ ರೀತಿಯಾಗಿ ಪ್ರೊಡಕ್ಷನ್ ಆಗುತ್ತೆ ಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸ್ ಎಲ್ಲಿ ಪ್ರೊಡಕ್ಷನ್ ಆಯ್ತು ಅನ್ನೋದನ್ನ ಸ್ಟಡಿ ಮಾಡಿದ್ವಿ and the female reproductive structure and the how the female gamete is produced so formed that we have studied. So in the next class we are going to study about the pollination ಯಾವ ರೀತಿಯಾಗಿ ಆಗುತ್ತೆ ಫರ್ಟಿಲೈಸೇಷನ್ ಏನಾಗುತ್ತೆ ಸೊ ಪೋಸ್ಟ್ ಫರ್ಟಿಲೈಸೇಷನ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅನ್ನೋದನ್ನ ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸ್ ಅಲ್ಲಿ ನಾವು ಸ್ಟಡಿ ಮಾಡ್ಲಿಕ್ಕಿದ್ದೇವೆ. ಓಕೆ, ಸೊ thank you dear students. If you have any doubts you can write down in the comment box. So ನಿಮ್ಗೆ ಅರ್ಥ ಆಗಿದೆ ಅಂತ ಅನ್ಕೊಳ್ತೇನೆ. If you have any doubts you can write down. Okay, thank you.